Hi, welcome to the Bioinformatics Chat. Today I'm speaking with Filippo Castiglione. Hi, Filippo.
Hi, Roman.
So, Filippo, I know you from your work on modeling the immune system, and this is what I want to talk to you about today. I have to say that this topic is very apropos for me right now because both my son and I have a cold, so the things we'll probably be discussing are quite literally happening inside me right now. But what is your background? What brought you to study the immune system?
So I'm a computer scientist. It just happened by chance that I met Franco Celada at the time I was choosing for my baccalaureate thesis at the Department of Computer Science in Milan a long time ago. I was interested in artificial intelligence and this kind of things, which are the usual things people get, enthusiastic about when they are computer scientist students. But then I met Celada, and the whole project about simulation of the immune system looked like an artificial life kind of, paradigm to explore. So I like that very much. So I started to, be involved in the discussion, and in the end, I accepted to do my baccalaureate thesis on this topic.
So how long did it take you as a computer science student to really get comfortable with the immune system? Because I hear from medical students that immunology is one of the toughest subjects they have to master. So
yeah, it took a long time because the real problem was not just the complexity of the immune system, because as a computer scientist, we somehow are used to handle with complex system. The point is that the language which is used is completely different, and the background is completely another type of background. So it was basically impossible to follow and to learn things in immunological books, for example, because of all those experimental details and so on. And also the mindset is different because they do not come from biologists. They are less quantitative. They tend to be approximative most of the time. So we are used to think and speak and reason in a different way. So this was a major problem when I first started. But with the years, I learned to appreciate the other way of thinking, because it's the only way to cope with this enormous complexity. If you stop and look into the details at every single bit of information, you'll never reach any conclusion. So you have to do some big leaps. And so I learned to do these big leaps myself, but it took time. And of course, the vocabulary is different, and so on and so forth.
Yeah, that's a really good point about the language and vocabulary, and in general, this very qualitative description of this process. And I have to say that I actually learned a lot about the immune system just by reading your papers and Celada's papers, which make a lot of these things much more precise. And of course, this is still an approximation. This is still a model, but it really sort of allows you to focus on the essential interactions between the various agents there. And so when you met Celada and started working with him, what year was it?
It was 92.
Oh, it was 92. So that was exactly the year when Celada's paper was published. And
yeah.
So I'll link to the paper in the show notes, but this is, I think, one of these classical papers that introduced a lot of the notions that we will be discussing today. That was Celada and Seidel. Is that right? Seiden, yeah. So a computer model of cellular interactions in the immune system. Are you familiar with the history before that paper? So I understand that before that paper, people used to model the immune system in terms of differential equations, probably because the computers weren't as widespread back then. How did those models work, and do they have sort of a reason to exist today? Can we apply them today, or are they completely obsolete?
Yeah. So the point is that what Celada and Seiden did was completely new at that time, and it's still an approach which is followed by not so many people. The point is that in the research in theoretical immunology was at the beginning, and people were facing a number of different questions. So with the usual or classical way of modeling things, they were writing system of differential equations, for example, to solve or to understand a specific piece of the whole picture. Now, the novelty of the approach of Seiden and Celada was that they tried to build a model of the immune system in its whole complexity, entire complexity, of course, in a very approximate way, but they tried to put the main ingredient of the whole soup. This was completely novel, and it was possible basically for a combination of reasons. The first, as you said, was the computational power was, reached a certain level. Previous work have identified the concept of diversity as a very important in the immune system, and there were already models, in particular of Packard and Perelson and remember the third guy. They published a paper in which they used binary strings to represent the specificity of the receptors in cell surface of lymphocytes, right? So this concept was the main concept that Franco Celada and Phil Seiden borrowed from existing literature, and they implemented something on powerful computers in a way which was novel. So the third thing which was important in this process was the fact that Phil Seiden, as an astrophysicist, was used to handle this complexity in a discrete way by using the concept of cellular automata, which was also somehow new at that time. So he built, with his knowledge, a model of the immune system, the way he used to build models of galaxy formations. And he was guided by Franco Celada, who was an immunologist, who spent a lot of years at the age of theoretical immunology. So he was pushing new concepts into Phil Seiden's programming while he was programming ImmSim. So this is the way I see the whole genesis of this thing.
Very interesting. So give us a general overview of Celada's and Seiden's original model. What are the main actors in the immune system that they try to model, and how do they interact?
All right. So at the time they built this immune system model, few concepts of immunology, theoretical immunology, were still, under debate. For example, the fact that the adaptive immune system would benefit from cell cooperation was not to be taken for granted. I mean, it was something new, so the role of antigen presenting cells, for example. And the other thing was the theory of clonal selection by Bournet has already been, let's say, enunciated, but there were no model somehow empowered in its complexity. And so they, taking this idea of modeling with binary strings the receptors, let's say, chemical-physical properties, they were able to implement a model of the clonal selection in which many different pathogens could have been modeled at the same time. So it was general model, while the rest of the immune system model at that time was basically writing an equation for a specific pathogen. They were not able to accommodate for more than one. This means basically that those models, equation-based, were not able to cope with the concept of cross-reactivity. So this was instead one of the major feature of the Celada-Seiden, who implemented therefore a population of lymphocytes of different classes. There were B cells and T cells and professional antigen-presenting cells like macrophages. And these cells were all equipped with the specific receptors, which then was selected by the specific pathogen they were injecting artificially into the system. So this was the novelty, being able to embrace the complexity of immune response somehow in a more comprehensive way, I would say.
Right. So in Celada's paper, we have 3 types of cells, right? You mentioned so B cells, T cells, and antigen-presenting cells. And the pathogen is modeled as just antigens, and there are also antibodies. How do these agents interact with each other?
Right. Okay, so the model is a cellular automaton, or it's an extended version of cellular automaton, because it's probabilistic first, and secondly, the concept of neighborhood of a certain lattice site is a generalized version of it. So some people call it hypercellular automaton. Nowadays, people call it an agent-based model. Okay, so basically, you have a discrete description of what a cell is. It's an agent, okay, and this agent is free to move on a certain description of a volume or the space, and this agent is free to meet and interact with the other, okay, with other cells, which are other agents. Now, this interaction is made in 2 ways. If it's specific, it is made through the binary string matching, which is reproducing what happened in reality when 2 molecules bind together. This is by immunology is called forming the immunological synapses between 2 cells. So they get in contact, their receptor shake hands, they bind, and then other molecules bind on the 2 surfaces, between the surfaces of these 2 cells. And while they are bound, they signal each other through cytokines and this kind of things. So this biological phenomena were implemented in a very, let's say, of course, approximated way in the Celada-Seiden model through this concept of binary strings matching and with probabilistic events. So when the match was perfect between 2 strings, then the probability of having a follow-up event would have been very high. But the match could also be unperfect, so not perfect, and this would be equivalent to a smaller probability to interact. Okay, so all this was at the core of the model, and therefore the execution, a run of the simulation would have been the equivalent of a series of stochastic events moving the system from one state to another state in a stochastic way.
And so this was Celada's paper, but that year you also wrote your bachelor thesis with Celada. So what was your contribution back then, and how did you evolve the model in the next 25 years?
Yeah. So at that time, the model C-ImmSim was written in APL, which was an IBM proprietary language, which was very complex. Seiden was a real expert in APL, but it really prevented, it was clear even in their mind that it prevented the dissemination of this model. So my contribution, the first contribution was to implement a specific function in APL, that was my bachelor thesis, to model the hypermutation phenomena, okay? So with that, I did my study and I closed it there, my bachelor. But later on, I decided to recode the whole ImmSim model in C language. So I did it. And in the year after, I just moved on improving it and customizing the model to accommodate for specific pathologies and this kind of thing. So my contribution started as a mere porting of the code, and then evolved in my, I would say, main scientific interest, which was applying the model to different pathologies. Yeah, and of course, in the meanwhile, I was also concerned with the performance and this computer science thing, like having a simulation, being able to run efficiently and increase the space, increase the number of cells, and increase everything. So this is what I did later on.
Yeah, and you mentioned the APL or APL2 programming language, and I think many people just don't appreciate how sort of weird and exotic that language is. And I say this as a fan of exotic languages myself. So I imagine it was quite overwhelming for you having to learn both the basics of immunology and the APL language. I think most programmers and computer scientists have barely heard of APL, let alone know it. So yeah, what was it like for you to dig into this APL code base?
Yeah, it was really a nightmare. I mean, I was too young to be able to, take it just as a challenge. And of course, you can imagine as a bachelor student, you have limited time. So I couldn't spend too much time learning APL, but likely there were other people around me which helped me in writing this piece of code that I had to write. And in the end, it wasn't too long. I mean, although APL is a very compressed language, so that even 100 lines of code can do unbelievable complex things, I had a hard time writing this 100 lines of code, but there was a person who helped me in this, so I wasn't alone. And it has been very difficult, because as you said, I had to understand immunology. And actually, I proposed to implement this specific biological phenomena of hypermutation. So I had to pick this specific thing and implement it in architecture which was very complex, because the system itself, once I translated in C, it was the equivalent of 20,000 lines of code, more or less. So in APL it was shorter, but APL is really complex. So it is exotic as a language. You have to imagine that you need a special mask to put on your keyboard, because it uses special characters. So that's really crazy.
So let's talk about hypermutation. So I think hypermutation refers to the mutation of B cells and probably T cells as well. And there is a lot of biological complexity to it, right? There's this VDJ recombination and stuff. Like, how closely do you try to model that phenomenon? Do you just generate random bit strings, or do you try to model the whole genetic, recombination and mutation?
No. There's no genetics in ImmSim. I mean, the molecules are binary strings, and as such, they are treated during all simulations. So the hypermutation was basically the random flipping of one bit in the paratope of the B cell, which changed his ability to recognize a certain pathogen. So my interest at the time was to to model a virus which was mutating on its own, and to understand how a permutation would have speed up or increase the ability of the immune system to catch this changing mutation. This changing antigen, like HIV. And in fact, starting from there, I later on studied the HIV, which had the same dynamics, or let's say characteristics in terms of a dynamics problem.
So it was never considered a problem that this sort of naive mutation may not be biologically realistic, and maybe you would get like more realistic, more precise results if you try to reproduce the biological processes related to mutation more accurately.
Yes, absolutely. I mean, we never reached that details of having realistic genes, for a reason which is very simple, that the information is too huge to be represented. I mean, although agent-based allow you to follow only those variables which are involved in an immune reaction, the model still has to represent the whole repertoire. So if you want something which, starting from the genetic level, reaches the real complexity of an immune system, it's not possible with the computers, even giant computer like those which are able today. So once you move into the space of binary string, you have a convenient way of representing information, and you can shrink the whole space to reasonable numbers so that your computers are still able to run the simulation. And also, I mean, if you want, you can always say that by increasing the binary string, you can always increase the real complexity. You could, in principle, in theory, reach the real complexity of the immune system, but you will never do, because you would need too much computer power than you can possibly have.
So in your papers, I saw references to both C-ImmSim, which is presumably ImmSim written in C, and also PAR-Im, which is presumably a parallel implementation of this. So what is the relationship between these two things? Are they different versions of the same program? Are they different programs? What is their current status?
So PAR-Im was basically the implementation of ImmSim in MPI. So actually, first we implemented with PVM, which was, let's say, a library to implement message passing in C. And I did this thanks to the help of Massimo Bernaschi, who was at that time working for IBM and developed the IBM version of PVM. So for him, it was an exercise. For me, it was an exciting challenge to start programming a parallel software. And but basically, the relationship between C-ImmSim and PAR-Im is that PAR-Im is C-ImmSim plus all code which is needed to exchange messages between processors. So it's the same, basically, piece of code. Later on, I had to abandon the parallel code because it was too difficult to cope with the implementation of biological details and the implementation of the parallel code at the same time. It was too difficult. So I decided that I wanted to focus on the biology, and I just left the computational complexity aside Although later on I came back to that task many times, and for example now the ImmSim version is multi-thread, so it's able to, it is parallel somehow. It's able to exploit the fact that modern processors have more cores, so basically they are parallel machines. So this is the relationship between the two things.
Yeah, I think we should clarify for listeners, as you many don't know, but PVM, I think it stands for Parallel Virtual Machine.
Virtual.
And MPI is a message passing interface, and basically MPI was used. I think it's still used nowadays somewhere, but it was used before as a way to implement parallel software that ran on multiple computers simultaneously, right? Whereas right now we've got more powerful processors, we've got more RAM, and so nowadays parallel programming usually boils down to just parallelizing within a single machine, right? So you move from the architecture which was distributed over multiple machines, maybe in the cluster or in a grid, to the architecture. Right. It's still parallel, but it's confined within a single computer.
Right, exactly. Yeah, the only things I would add to what you said, which is perfectly correct, is the fact that while MPI, but in principle PVM as well, allowed the process to be split on different machines with different memory, where the current version of ImmSim, which is multi-thread, inherently has to use the memory which is on the same machine. So this is called shared memory paradigm, which is different than distributed memory. But perhaps these are details.
A couple of things about the model itself. So first, I have to disagree, or at least argue with you and Celada and Sidon about calling this model a cellular automaton, because and I think Celada and Sidon themselves point to an example of Conway's Game of Life as a sort of classical and well-known automaton. It's basically a grid where each cell is a living thing that can either be born or die depending on the number of neighbors that alive neighbors that it is surrounded by. And I think the core difference between something like Conway's Game of Life and ImmSim is that in ImmSim, there are cells or molecules which migrate from one cell to another. And therefore, you have this invariant that if something leaves a cell to a neighboring cell, it can only leave to one neighboring cell, right? Which sort of contradicts this paradigm that the next state of a cell is determined solely by its neighbors, right? So if you imagine two cells, A and B, both neighbors of C, and C currently has some kind of cell, like an immune cell in it. So C may migrate either to A or to B, but not simultaneously to A and B. That would be reproduction, right? But assuming it doesn't reproduce, it just migrates. And so because A and B are not necessarily neighbors themselves, right, they do not know how to evolve themselves, right? They have to depend on C supplying this token either to A or to B. And I think this is a pretty important difference between cellular automata and this. I think I like the agent-based model more because it underscores that we're modeling the agents themselves, so the cells and molecules, and not necessarily the cells in which they reside. Cells, the sort of grid cells, right, or these squares. Sites, yeah. So, and I think you even wrote something about efficiency there, right? So in order to make this more efficient, you had to move to modeling the agents themselves and not sites, because keeping track of all sites was not memory efficient. Is that right?
Yeah. Yeah, what would you say? I perfectly agree with you. And I remember I had a discussion with Phil. But the point is that the view of serial automata has changed from those years to now, and they have evolved into something which are now called agent-based for this reason, because many of the constraints in the definition of serial automata were actually removed, and people were writing algorithms in the way they liked it, and so nowadays people call agent-based or serial automata the same thing, taking for granted that the strict definition of serial automata does not apply anymore. I mean, no one uses life to make a model of biology or physics, whatever, anymore. I mean, they will certainly change something in that definition. So the concept of neighborhood and so on and so forth. There is one discrete model which is probably more close to this C-ImmSim vision of serial automata, which is the lattice gas. In a lattice gas, you have more particles residing in each site, while in a serial automata, you would have only one point, only one cell in a site. So diffusion and interaction would overlap somehow. It would be difficult to, say, to describe in those formal terms what was diffusion, what was interaction. But in the lattice gas, there were two different steps of the algorithm. There was a moment in which cell would diffuse, and there was a moment in which cell would interact. And interaction was local in each site. So this view of building discrete modeling, it's very much in line with the general paradigm of serial automaton. And in fact, Wolfram, who was one of those who made serial automata famous, he wrote a book with the title "A New Kind of Science." So it was a shift in the paradigm in general. So it's better not to be too much attached to the definition, because what was very powerful was the idea that you could model things using a computer rather than solving differential equation, which was the way to do until that time. So now we have two ways of simulating reality. One is by solving differential equation with the numerical schemes, and the other one is to representing thing in a serial automata fashion or in an agent-based fashion. Those are two, let's say, complementary paradigm in simulation now, in physics, in biology, in every complex science, basically.
Yeah, so one of my objections to calling this a cellular automaton is, as you said, just based purely on the mathematical definition. But there's also a second one, which is by moving from differential equations, which is a completely continuous model and completely sort of aggregate-based model, to cellular automata, we introduce not one but two discretization steps. So we have discrete agents which move between sites, but also the sites themselves are discrete. And I think this second discretization step, it seems to me as completely superfluous, right? If we have discrete molecules and discrete cells, but they can perfectly well have continuous coordinates, and they can move like based on some kind of gas model. They can move continuously, and still we have a discrete model, and we can model all the discrete interactions. So is there any reason to have these discrete sites and not just assign numeric coordinates to the agents?
No, the only reason, I mean, you're perfectly right. I can tell a couple of things. The first is that agent base of today are not restricted to a discretization of space. As you said, they have continuous coordinates. And in fact, many models represent cells as volumes in their coordinate, and they are much better in representing, for example, the forces which push one cell against the other and to create tissues, and they are very nice models. So, true, I mean, you can't call those agent-based cellular automaton because they are too far away from that mathematical definition. The second thing that I want to say is the fact that at the time Seiden built this model, he was somehow inclined to use a discrete space because he was coming from modeling of galaxy, very similar to biological tissue if you want, and he was using a language, APL, in which vectors and arrays were the main data structures. So it was much easier to code this common complex process with discrete space rather than using, differential equation. That's a really complete different way of working. And this allowed to build quite sophisticated simulation, yet constrained by the memory and the computational power of that time. So discretization allow you to, to save memory somehow.
But again, you can discretize the system in two different ways. And even if you use an array-based programming language like APL, you can still have arrays of coordinates and arrays of velocities, and it's still perfectly suited for array manipulation without discretizing the space itself.
Yeah, but if you discretize the space, you eliminate the problem of understanding who is interacting with who. Or you somehow move, from the modeling point of view, you move the problem of deciding who is interacting with you by the problem of determining how close two objects are. You move this problem to the problem of just throwing a dice in a probabilistic Monte Carlo way of simulating things. So it's a modeling choice somehow. Rather than, computing who is closer and deciding who has to interact and who should not, you make everything, you give all cells which are on the same lattice site the chance to interact with a different probability, and you somehow rank the probability in a kind of a uniform way. Now, in the first case, with the real coordinates, you would ranking according to the distance, for example. The closer, more likely to interact. In ImmSim, you don't have this. It's like if you have same equidistance, so you give all of them the same probability. I think Seiden and Celada were more concerned about the logic of the immune response rather than the physics of the diffusion and this kind of physical kind of process. They were more interested in understanding which receptor was the one who would have emerged as the useful one, So they just disregarded these aspects. I took it this way.
Yeah. And you made an excellent point that if you have continuous coordinates that you just hold in an array, then at every step determining which agents are close to each other and which agents should interact. So that becomes a quadratic complexity problem because you have to analyze order of n squared over 2 distances, compute them, right? And I imagine in order to find that, you would use some kind of clever data structure like KT3 or something like that. So so you said that right now you're modeling coordinates as continuous. So do you use any of those tricks to find this quadratic complexity, or how does it work in your model, in your current model?
No, actually, I don't use continuous coordinates. I still have discrete coordinates, but for computational reason, makes my life easier because I learned that the biology of the problem have more meaning. I mean, in the future, if I had a ball, a magic ball, I would, of course, love to build an ImmSim version in which the topology plays a very big role. But this leap is too big. It's gigantic. There's so much complexity to take into account when modeling tissues and changes in the, permeability or whatever, diffusion and density, which it's just beyond my ability. And as a matter of fact, I don't know of anyone who is able to make an immune model with such complexity. So I focused on other more biologically oriented problems or disease-oriented problems rather than this technical aspect, which I'm sure sooner or later will be solved, but on very large computers by people who is definitely more skilled than me in building complex models.
Yeah, fair enough. But I can't resist to interrogate you a little bit more about this technical issues. So you mentioned the topology, and this is another thing that I was thinking about, because I think in one of the papers you say that you model only like 1 cubic millimeter or a couple of cubic millimeters of the lymph node space, right? So we're not talking about like blood and lymph vessels and capillaries. It's just some space where all the agents can meet. And so I have two questions regarding this. Well, first, I think you apply your model to modeling like whole organisms. For example, I saw your recent paper where you were one of the co-authors about a vaccination of sea bass.
Mm-hmm.
So there you or the primary authors just apply this model, which models essentially like a tiny volume of the lymph node, to the whole organism, whole fish. And so how realistic is that? Because clearly when you try to model the immune response and the result of this vaccination in this big organism, clearly those things, they must matter a lot, right?
Yes. No, that's exactly the point, which interests me very much. It's so, as you said, ImmSim at the origin, but even, let's say, today, represent a portion of the lymphatic system in general, without specifying what it is. Certainly it includes a piece of a lymph node. But not only that, because you have the presentation of the antigen, which takes place in the lymph node, but you also have effectors activities, immune system effects in the simulation. So this means that you are representing also some tissue, okay? So the volume is the mathematical representation of a piece of a body. It doesn't say which piece. It could be all body compressed in one piece. Yeah. So you are neglecting all the diffusion, all the trafficking, everything. This is what the model is up to date. But I understand that there are a lot of problems connected to the migration of cell, what it's called cell trafficking. The timing of the response is very much dependent on how efficient this trafficking is. And in fact, if you have a person, a patient which lies on a bed, his immune system is much weaker than someone who moves and do sports, just because the trafficking of cells in the channels and in the tissue is much more efficient when you move because of osmosis in the lymph channel. So for this reason, I am trying since a few years to extend ImmSim to cope with realistic anatomies. That's why we developed a multi-thread version in which we can have more compartments, let's say. and we have the trafficking of cells between compartments. In this way, we could represent a lymph node. Now we have the computer power to represent the whole lymph node, plus a piece of a tissue. For example, a skin or, another place where the antigen infects, for example. Now, this is what I want to do, but I'm facing a lot of problems because there's knowledge missing there. There technicalities to be faced, how long it takes a cell to go from point A to point B, and all these kind of things. But once we will solve all this, then it will be just a matter of computational memory and speed to implement a realistic anatomical representation of a body. We try to start from NMR data to reconstruct the lymphatic structure, but we learned that it's impossible to know where the vessels are just because they are too small. There's no detection imaging technique which is able to tell you where the lymph channels are. So you can, in principle, know where the lymph nodes are, but you don't know how they are connected. So this information is useless. Either you guess, you construct a network, or you can't do these things. So I agree with you that this is, I think it's probably the future of the immune system simulation, immune system modeling. One possible way, which is what I call the anatomical detailation, how would you say, specification of details of the anatomy. There are other ways to enlarge this, which is the molecular way, but that's another story.
So you mentioned modeling different compartments in connection to the multi-threaded implementation. Is that the case, that you map each compartment to its own thread?
Yes.
Mm-hmm. And in this case, when you have like either no topology at all or this very simple topology of several compartments that interact with each other, I'm wondering how much benefit does it bring to model the space at all, the spatial coordinates? So one alternative way that I can imagine is just talk in terms of probabilities. So if two cells are in the same compartment, they have the same constant average probability of interacting. And so you could replace modeling. Like you said, you were not interested in physical details, so you can go even further and just say, I don't know where these molecules are, but I know that on average they have this probability of interacting, of like meeting within the space. So did you consider that?
Yeah, but this is exactly the kind of consideration we do when we, write a paper and we have to explain to the referee that we are representing a piece of the whole body. So we just say we are compressing the whole body in a place, and then the problem is to determine how likely a cell meet in the same point. But on the other hand, we also understood that spatial effects are meaningful. For example, when you simulate a viral infection, one thing is to have the viral spread all over, so all over your volume or your space, so that the probability to be found and all the other events which are necessary to bring to the immune response, which also depend on cell meat, are very likely. And the other thing is to inject the virus in a single point, maybe in a place where immune cells are not so, present.
Yeah.
So this would, in certain situations, would be the equivalent of giving, a certain advantage to the virus, which could make a difference between life or death. So spatial compartmentalization of tissue plays a big role in the immune response. And I think these are, I'm very curious to really simulate these aspects. I want to simulate an infection in the skin, and that everything which takes cells moving from the lymph node to that point and going back to the lymph node to present the antigen, all this machinery, which takes time because it may make a difference. And if you think at cancer, for example, the concept of metastasis, it's very much linked to these problems. The immune system has difficulties in coping with something which moves around and escapes. There are immunological sanctuaries, places where the immune system can't go or goes very, rarely. This kind of topics, I think, are very, very much influencing the outcome of a disease. It might not be, theoretically, in terms of theoretical immunology, very relevant, but it's very relevant in terms of the outcome of a disease. So medically, it's more relevant. That's what I want to say.
Yeah, the virus example is a very good one, very convincing. So when you model the virus infection, do you model like the virus particle infecting one cell and then incubating and the burst? Do you model all those things?
Yes, exactly.
Mm-hmm. Cool.
Yeah, no. So there are two types of this incubation can bring the cell to burst, as you said, which is it happens for certain viruses. Now, Ebola, I guess, but there are also other possibilities like HIV, which buds outside the cell, so that the cell doesn't die, but it keeps, producing virus all the time. No, I have both these two mechanisms, and it's nice to see that it really makes a difference in the outcome of an infection.
Mm-hmm. Do you also model, like, can an infected cell present an antigen to maybe a killer T cell?
Yes, infected cells are equipped with the class 1 HLA molecules, so they present it. So all those things like immune escape is taking place in simulation. If you have a virus which mutate, it might acquire a mutation which prevents it to bind to the HLA molecule, for example. So it will basically become invisible to the immune response. Yes.
Very cool. Very cool. So it looks like your model can model a lot of things by now, but was there any time during this long history that you actually simplified something, that you decided to maybe remove some biological phenomenon because it turned out to be either false or not important for the simulation?
Well, I choose to, since I'm a computer scientist, my approach is to add the complexity, as you can imagine. I see C-ImmSim as an architecture, like an operating system, which has many things, many components. Now, these components can be, of course, commented out in your source code, or even, skipped with an if statement. So my approach is, let's put this new thing inside and see how it fits with the overall thing. Now, sometimes it happens that I add something which brings a lot of problems, as you can imagine, parameter tuning and everything. I use it, and then I decided not to use it anymore, for example. This happened with the hypermutation, for example. I don't use it often. I sometimes use it, but it's there. It's still in the code. It's still in this operating system. I can use it or not. So to answer your question, I would say yes, sometimes I just skip things, but this does not prevent me to implementing it, an approximate version of it. You know what I mean?
You never remove code, you just put it into an if statement.
Yeah, something like that. Or I bifurcate the version of the code, because I know that in previous version I had that code, and I can always go back and grab it if I want. So somehow I remove it, but it's like if it's still there somehow.
Yeah.
In my mind, it's still there.
Well, I imagine it should be in the version control, right? So you can always go back and find it.
Yeah, something like that.
And my understanding is that you also have like a domain-specific language for describing various new cells or new molecules and their interactions. I was reading your 2001 paper, and it has the snippets of code like interaction between B and TH, specific yes, match, condition, action. So that looked like a domain-specific language to me.
No, that kind of pseudocode was to express the fact that there are templates in the code that you can use. So you don't have to rewrite everything. You can cut and paste a function and change it to customize as you want. So you are somehow halfway in writing the whole thing from scratch. You don't have to rewrite everything. But on the other hand, this approach is not so straightforward to generalize.
So is this snippet of code that I see in the paper, is it like a direct quotation from the source code, or is it you sort of paraphrasing the C function? Or is this a piece of executable code?
No, I think it's more, I paraphrase the C function. I somehow simplified. I removed some complication, probably. But my experience, because I had many students in these years that have used the code, and my experience is that after I explain them what are the main data structure of the program and the logic in adding new entities, they can very easily go on by cutting and copying and changing these functions. They are able to do it quite easily. Of course, another thing is to have the overall view of the whole code, but they can definitely focus in the interaction only if they are interested in biology and implement new functions, biological functions. All the rest might be technical details. They can, leave it aside. That's my experience with them.
If a student wants to implement their own small immune system simulator, maybe over a weekend, where would you advise to start? Like, what's a minimal model of the immune system that is still interesting? What are the crucial agents and interactions to include there?
I think I would use the pathogen, of course, the B cells, if you want to produce antibodies, the T cells, if you want to implement intracellular pathogens like viruses. And then you need the T helper cells. So you need at least these three things, and perhaps macrophages. So four types of cells.
Mm-hmm.
So with B, T cytotoxic, T helper, and macrophages, or dendritic cells, one antigen presenting cells, let's say, you could have the adaptive immune response. Another thing is if you are interested in simulating the innate immunity, for example. In that case, you don't need B, you don't need T helper. It's much easier, let's say.
Mm-hmm. So what would be the simplest test that your immune system model is working correctly? So I imagine you would want to model like maybe secondary immune response, or what would be the main test?
The secondary immune response is usually the main test that your adaptive immunity is working properly, developing memory. And another test I usually do is cross-reactivity. This because I have cross-reactivity in my model, but you might not need if you want to make a really very simplified version of an immune system model. You can represent only one receptor and one type of pathogen. But if you want something, more generic and you want to model more infection at the same time, then you need to have more diversity, more receptors, and then you get the cross-reactivity concept, something which can recognize virus 1 and also virus 2, but with a less with a lower affinity, for example. Now, this kind of experiment to understand how cross-reactivity is working in your model is also interesting.
Cool. Okay, I think we'll be wrapping up. Is there anything that I didn't ask you about that you would like to talk about in this context?
Yeah, because I spent some time to develop C-ImmSim in a way that not even Celada is enthusiastic about, because it's very, very advanced, but many other people recognize it's very useful. And actually, I got in contact with people from pharma industry, which are definitely more into those direction. And it's the following. Since the time, the 90s, so the time of those immune system simulation, Celada Inside and model, etc., let's say from there on, bioinformatics has literally exploded, right? So it's a new, complete different scientific area, I would say, okay?
Yeah.
Okay, so which is probably running much quicker than mathematical biology in general, or the simulation of the immune system. So there is much more interest there, much more knowledge. Many tools have been developed. So that my point is, why don't we join these forces? Why don't we join these tools and we use the simulations we have developed with the bioinformatics tool and do some unified tool with which people can experiment immune system responses to a pathogen which is not represented with binary string. So I spent some time on it. I met people which are very good in developing prediction tools, peptide prediction tools, binding areas of the antigen prediction tools, and this kind of things. And with them, I developed a customized version of C-ImmSim, which is able to represent the pathogen not with binary string, but with the amino acid strings. I think this is a giant leap into the field, but there are, of course, huge problems which are connected with this prediction tool, with their, efficiency, or they're not able to predict everything with the 100%, of course, precision, okay? So the prediction that C-ImmSim will give when you run a simulation is not so high. That's the point. You can't point your finger on a specific peptide of the HIV as the one on which you should build a vaccine, because your way to discovering that peptide is not too correct, okay? But again, I am a computer scientist, so when I look at this problem, I see what I can do, and from my side, I can prepare the architecture to host new prediction algorithms in a very plug and play way. So I did this study, which has been published years ago on PLOS Computational Biology, which has been, let's say, welcomed by some people, but not too many as I expected. But at the same time, I believe that more and more people is moving into that direction. So sooner or later, they will realize how important was this work. At least this is a hope.
Ah, interesting. Yeah, so I think this podcast may contribute to spreading this idea and awareness of it.
That might be cool. Actually, there is a server, I could give you the web address of it, in which people from all over the world can run simulation of C-ImmSim, and by putting specification of primary sequence of a virus or a bacteria or a vaccine, and perform immunization experiments. So it's a live simulation which is available.
Interesting. Yeah, we should put it in the show notes on the website. And continuing this theme, what is the current relationship between you and Celada? Does Celada still work in this space, and do you compete? Do you collaborate on this?
Yes. I mean, we are writing an article together, and He's a real scientist. I mean, at his age, he has the same enthusiasm as when I met him. It was decades ago, and he's a great scientist. So it's really, I consider my scientific father somehow, because the thing I have done have been under the umbrella of this immune model. Let's say many things I have done. I did also something else, and it's always nice to see how his view of the model itself. So at the beginning he was at the beginning he was working with Phil, but from a certain point because Phil left us prematurely, Celada started to collaborate with other people, with another Italian guy who developed this version of C version of ImmSim, starting from pieces of my code, and other people's also have developed another version of C-ImmSim, thanks to my code somehow. And so Celada collaborated with one of this group first, and now he's collaborating with me, but he still, has this will to exploit this computer model.
Cool. Yeah, well, Filippo, it was very interesting to talk to you. Thank you for coming on the podcast.
Thanks to you, Roman.